So far, we have seen double fertilization. Double fertilization is a special character in angiosperms. During double fertilization, the zygote is formed. In addition to zygote, the primary endosperm nucleus is formed that later on gives rise to endosperm. So, after fertilization, the endosperm and zygote are formed. Now, we shall see post-fertilization events. As the name indicates, post means after fertilization. After fertilization, what are the events that takes place? Is post-fertilization events. That is, all the happenings of after fertilization are the post-fertilization events. In angiosperms, after the fertilization, the endosperm is formed. The endosperm always formed after fertilization. The embryo is also formed. But in angiosperms, the endosperm develops first, followed by the development of embryo. It means the endosperm is first formed, then the embryo develops. The ovules develops into seeds, whereas the ovary develops into fruit. All the above events are together called post-fertilization events. Now, we shall see one by one what happens after fertilization. After fertilization, the sepals, that is the calyx, sepals fall off or wither off. Similarly, the petals also fall off. Even the stamens also wither off. The first happening. So, after fertilization, sepals, petals, and even the stamens wither off or fall off. The ovules present in the ovary, they are the fertilized ovules. These ovules develop into what are known as seeds. So, after fertilization, the ovules develop into seeds. The embryo sac contains antipodals. Three antipodals are there at the chalazal end. What happens to the antipodals? The antipodals after fertilization disintegrate, or you can say disorganize. The egg apparatus consists of egg cell and two synergids. See what happens to these two synergids. The synergids also disorganize or disintegrate. That's all within the embryo sac. What happens to the integuments? There are two layers are there. Outer layer of integument and inner layer of integument. Or inner layer you can say. The outer layer of integument transformed into what is known as seed coat. Outer layer is a more thick layer. It is called testa. What is testa? The outer layer of seed coat, which is nothing but integument. Similarly, the inner layer of integument also transforms into another layer called tegmen. So, the integuments after fertilization develops into seed coat. Outer layer is called testa and the inner layer is called tegmen. Okay. What happens to primary endosperm cell? PEC. Primary endosperm cell develops into what is known as endosperm. That is, the nucleus of this cell is triploid, which divide and redivide forms the endosperm. The endosperm nourishes the developing embryo. So this is the reason in angiosperms the endosperm is first formed, then the embryo is formed. It means the embryo is nourished by the endosperm which is already present. What happens to the zygote? The zygote is a diploid one. The zygote divides and redivides and develops into an embryo. So, ovary, ovary. The ovary is nothing but the basal part of the pistil, which contains the ovules. This ovary, after fertilization, develops into a fruit. Okay, so that's all. Now we shall see the development of endosperm. We know that after fertilization in angiosperms, endosperm is formed. How this endosperm is formed from the primary endosperm cell? It is nothing but the development of endosperm. Okay. First, we shall see what is endosperm or the definition of endosperm. Endosperm is the nutritive tissue which nourishes the developing embryo. This is the answer for what is endosperm. Endosperm is the nutritive tissue means it nourishes the developing embryo. When the embryo is very young, it supplies the food for the developing embryo. So that is the function of endosperm. Okay. Now, we shall see the development of endosperm. How the development takes place. After fertilization, the primary endosperm cell, PEC, its nucleus is triploid, which divide mitotically, divide and redivide many times mitotically, and forms a tissue called endosperm. The endosperm is triploid, a very important notable point for CTR need examination. Why the endosperm is triploid? Because it is formed from definitive nucleus, diploid, fuses with male gamete N, then 3N is formed. Or you can say it is the product of triple fusion. Okay. Thus, endosperm occur as post-fertilized tissue in angiosperms. A very important NEET or CET point. So the endosperm is formed after fertilization only. That's why it is called post-fertilized tissue in angiosperms. Contrast to this one, or contrary to this one, in case of gymnosperms, the endosperm is formed before fertilization. We call it as pre-fertilized tissue. It is one of the major difference between the endosperm of angiosperms and the endosperm of gymnosperms. A notable CETR NEET point. If we examine the cells of the endosperm, the cells are filled with reserve food materials. So the endosperm cells store lot of reserve food materials. This food material is used for the development of embryo. Okay. Now, coming to the development of embryo, there are three types of embryo development. One we called free nuclear endosperm type of development. Then cellular type of endosperm development. Then helobial type. Free nuclear, cellular and helobial type of embryo endosperm development. Okay. In your syllabus or in NCERT textbook, helobial type is not mentioned. Okay. Now we shall see free nuclear endosperm. How the free nuclear endosperm is formed. After fertilization, the zygote is formed, the zygote is diploid. Actually, at the center of the primary endosperm cell, vacuole appears. The vacuole grows. Now, the nucleus of the cell, that is pen, divide, redivide many times mitotically, thus form large number of free nuclei. Free nuclei means only nuclei are found. Here, the cytoplasm will not divide. Only the nucleus divides mitotically. So that's why the division is called free nuclear division. So the endosperm is free nuclear. In which plant we come across this type? Very common example is coconut water. Coconut water means the tender nut water. What we say? ಎಳನೀರು ಅಂತ ಕರಿತೀವಲ್ಲ ನಾವು, ಎಳನೀರು, that is a liquid endosperm. A very important, see it your neat question. So in this free nuclear type of endosperm, the nucleus of the pen divides many times and forms free nuclei, and it remains as free nuclear endosperm. Okay, this is one type of endosperm development. In some other plants, other type of endosperm development also noticed. What to say? Cellular type of endosperm development. So we shall see now that one also, cellular type. Okay. Again, coconut may be quoted for this. The tender nut, ಅದು ಎಳೆದಿರಿದ್ದು ಮುಂದೆ ಕಾಯ ಆಗುತ್ತೆ. That itself we call cellular endosperm. Example, white kernel of coconut. ಏನು ನಾವು ಕಾಯಿ ಅಂತ ಕರಿತೀವಿ, ಅದು cellular endosperm. Now, in coconut, free nuclear endosperm becomes cellular endosperm by undergoing cytoplasmic division. It means after free nuclear division, the cytoplasm also divides and the endosperm becomes cellular one. So thus we notice two types in coconut, free nuclear endosperm as well as cellular endosperm. Okay, so that's all about the development. Not much information is discussed with respect to this one. Next, what happens to this endosperm? The endosperm, as I told you, is a nutritive tissue, nourishes the developing embryo. Nourishes means supplies the food materials. That's all. See, in some plants like pea, groundnut, beans, horse gram, then the soybean, green gram. In these plants, the endosperm is completely utilized by the developing embryo. It means the seed do not contain endosperm. So, seeds do not contain endosperm. Such seeds are called exalbuminous seeds. Seeds without endosperm. In some other plants, the endosperm persist or remain in the seed. Even after the development of the embryo, the endosperm is persist. Very good example, coconut. ಏನು ನಾವು ಕಾಯಿ ಇರ್ತತೆ, ಅದು ಕಾಯಿ, it is nothing but endosperm, solid endosperm, we can say now. Okay, coconut, embryo ಚಿಕ್ಕದೊಂದು ಲಿಂಗು ಥರ ಇರುತ್ತೆ, that is the embryo. ತೆಂಗಿನಕಾಯಿನಲ್ಲಿ ಸಣ್ಣದೊಂದು ಲಿಂಗು ಥರ ಇರುತ್ತೆ, that is the embryo. ದಿ ವೈಟ್ ಪಾರ್ಟ್ ಏನು ನಾವು ಕಾಯಿ ಅಂತ ಕರಿತೀವಿ, ದಟ್ಸ್ ಆಲ್ ದಿ ಎಂಡೋಸ್ಪರ್ಮ್. ದಿಸ್ ಈಸ್ ದಿ ಎಡಿಬಲ್ ಪಾರ್ಟ್ ಇನ್ ಕೋಕೋನಟ್. ವಿಚ್ ಈಸ್ ದಿ ಎಡಿಬಲ್ ಪಾರ್ಟ್ ಇನ್ ಕೋಕೋನಟ್? ದ ಎಂಡೋಸ್ಪರ್ಮ್. ಓಕೆ, endosperm is also found in castor seed. Castor means outla. The seeds which contain all endosperm called albuminous seeds. Thus we come across exalbuminous and albuminous seeds. So this endosperm is also used at the time of germination of seed also. Okay. So that's all about endosperm, development of endosperm. Now we shall see the embryo. I told you, after fertilization, the diploid zygote is formed. What happens to this zygote? The zygote develops into what is known as embryo. Where is embryo? The embryo is present at the micropylar end of embryo sac. Okay. The diploid zygote or its nucleus divide mitotically and forms the embryo. Okay. By this time, the endosperm is already formed. It nourishes the developing embryo. Okay. If we trace the development of embryo from zygote to mature embryo, we call it as embryogeny. You know, embryogeny means the development of embryo starting from zygote to mature embryo. So next topic, development of embryo, that is dicot embryo. Okay, so this question so far it's not appeared in your examination, a very important question for 5 marks. Refer the diagrams, very important one. Okay, now we shall trace the development of embryo. The zygote is unicellular. You know, the ovum is unicellular. After fertilization, the ovum is transformed into zygote. Zygote is also unicellular. How this unicellular zygote develops into mature embryo? If we trace this, we come across the following steps. First, the zygote undergoes a transverse division and forms two cells, the upper cell and the lower cell. Or you can say now, the upper cell and the basal cell. The basal cell divide and redivide mitotically and form the basal suspensor. Suspensor is at the chalazal end. Now you refer the diagram, third and the fourth diagram. In the development of embryo, you can see the suspensor. What is the function of suspensor? It absorbs the food materials from the endosperm and also transport the food to the developing embryo. Okay, that is the function of suspensor. Now, the terminal cell divides longitudinally as well as transversely and forms what is known as proembryo. So the zygote develops into proembryo. In the next stage of development, the cells of the proembryo divide and redivide, develop into spherical shaped embryo, what we call globular embryo. Proembryo develops into globular embryo. Okay, see the diagram now, the globular embryo. In the next stage, the globular embryo again transformed into or develops into a heart-shaped structure. heart-shaped embryo. See the diagram, the heart-shaped embryo. Finally, the heart-shaped embryo develops into mature embryo. Okay, so that's all about the development of dicot embryo. Okay, so it is for 5 marks. Now see the diagram. See the diagram now. The A diagram shows fertilized embryo sac, showing the zygote. There is zygote. Where is zygote? At the micropylor end, the zygote is there. Then pen is there, primary endosperm nucleus, which is present in a big cell, primary endosperm cell. What you call previously the central cell. The central cell transformed into primary endosperm cell. That is the first diagram. Second, stages in embryo development, that is in dicot. Single cell zygote ಕೊಟ್ಟಿಲ್ಲ ಅದನ್ನ. Next only directly globular embryo is there. See the embryo at the top, globular. Top means at the basal region you can say the embryo. Globular embryo containing many cells. Elongated part is suspensor. So the third diagram, that is heart-shaped embryo. Okay. Then the last one, that is the mature embryo. Suspensor is very much reduced. So there is a radical, cotyledons, primule, the mature embryo. So that's all about the development. Okay. Now, we have seen the development of zygote and also the development of embryo. No, there are two types of embryos. Two types of plants are there, dicots and monocots. Now we shall see dicot embryo. See the diagram also. Okay. It's for 2 marks, you can say. Explain typical dicot embryo for 2 marks. A typical dicotyledon embryo shows embryonal axis. See the diagram. The embryonal axis with 2 cotyledons. Carefully examine the cotyledons. Cotyledons are very big in size. They are. Okay. Because these cotyledons stores lot of reserve food materials. How many cotyledons are seen? 2 cotyledons. Because it is dicot. A very important difference between dicot and monocots. The dicots always have 2 cotyledons. Monocot have only 1 cotyledon. Now, above the cotyledon region, we call epicotyl region. So, embryonal axis having epicotyl region. This epicotyl, as the name indicates, epi means above, coty means cotyledon. Above the cotyledon, the region we call epicotyl. This epicotyl region ends with what is known as plumule. Carefully observe a pointed structure in the embryo. What happens to this plumule? The plumule, when the seed germinate, develops into shoot system. See, shoot system means which is found above the ground. Shoot system. Now, the region below the cotyledon, you just observe the diagram. We call hypocotyl region. Hypo means below. Cotyledon means refers to cotyledon. So below the cotyledon, the region of hypocotyl is present. You examine carefully, which ends with what is known as radical. Radical or root tip. So embryonal axis consists of the upper plumule, basal radical, and in the middle cotyledons, two cotyledons. The plumule develops into shoot system, whereas the radical develops into root system. The root tip is covered with a cap-like structure, what is known as root cap. See the root cap also. Okay. So that's all about the typical dicot embryo. Very simple it is. Very simple. Okay. ನಾವು ಕಡಲೆಕಾಳನ್ನು ಅಥವಾ ಶೇಂಗಾ ಗ್ರೌಂಡ್ ನಟ್ ಸೀಡನ್ನು ಓಪನ್ ಮಾಡಿದರೆ ವಿ ಕಮ್ ಅಕ್ರಾಸ್ ದಿ ಡೈಕಾಟ್ ಎಂಬ್ರಿಯೋ. ಚಿಕ್ಕ ಪಾಟ್ ಇರುತ್ತೆ, ಕಾಟಿಲಿಡೆನ್ಸು ತುಂಬ ದೊಡ್ಡದಾಗಿರುತ್ತೆ, ಕಾಣುತ್ತೆ ನಮಗೆ. ಹೌದಾ? ಅಂದರೆ ಇಫ್ ಯು ಡಿಸೆಕ್ಟ್ ಎ ಡೈಕಾಟ್ ಸೀಡ್, we come across the two cotyledons and the embryo. Embryo means embryonal axis with the radical and plumule. Okay. So that's all today's class. Thank you.